In this video, I'll talk through the 2025 National 5 Biology paper and explain how I would approach each question. Starting off in the multiple choice with question number 1, we're asked about the structures that would not be found in a typical plant cell. Chloroplasts contain chlorophyll, which is involved in photosynthesis, which is something that would be required for a typical plant cell. Cell membranes control the entry and exit of materials, again something that plant cells would require. Plasmids are extra rings of DNA which are found in bacterial and yeast cells, not a typical plant cell. That's going to be our answer. Mitochondria is responsible for aerobic respiration for energy. That is something that a plant cell would require. In this next one, we're talking about osmosis and the movement of water when cells are placed into different solutions. To help us with this one, we need to remember that if there's high sugar content, there'll be low water content. This would be an example of a very concentrated solution. On the other side, if there was lower sugar, there would be higher water. A high sugar solution may also be described as being hypertonic versus hypotonic for low sugar. So now what I'm just going to do is label each of the pairs in terms of the lower and the higher water concentrations. Next thing to remember is that osmosis is going from high to low water concentration. So in the first one, that means the water is going to leave the cell and head into solution. That would lose mass, not gain. Not going to be our answer. Next up, within it's an isotonic solution, the same concentration in and out of the cell, nothing will change. Not going to be our answer. In this one, we would have water coming into the cell, therefore it would gain mass, and the last one, D, would also do that. When it comes to how much water would be gained, it's about the difference in concentration. D has a larger difference in concentration, meaning there will be more osmosis happening, and that cylinder would gain the most mass. In this next one, we are told that there are 34% of thymine bases. We have to remember our parents, where if within a section there's 34% of thymine, there'll be 34% of adenine, which gives 68% as we stand. The total percentage will be a 100, so we take our 100, take away the 68, leaves you with 32%. That means that our cytosine and guanine together equals 32%. So each of them will equal half of that. So each of them will equal 16, which looks like answer C for that one. Shuffling next, we're talking about an enzyme reaction of breaking down fats by an enzyme called lipase. pH was recorded in each of them. In which of the test tubes would the pH stay the same? Okay, what can we tell for this then? If reaction happens, then our pH is going to change. Okay, so milk has fat in it. Lipase is the enzyme that breaks down the fat. That means that R will work. In S, we're missing the enzyme. We've replaced the enzyme with water. That's not going to work because there's nothing there to break it down. And in this one, by boiling the enzyme, that is going to become denatured, which means its shape will have changed and it will no longer be able to work on the fats and break them down. So that is not going to work. Question is asking us, which ones would they stay the same? So we're looking for the two that do not react. That means that S and T is our correct answer because S and T, nothing would happen, therefore the pH would stay the same. Next up, we are looking at some blood, which is containing a chemical here, which is an amino acid, I'm pretty sure. What are we told? We're told that 100 grams would have 15 grams of that chemical. And then we are told we want 5 grams of hemoglobin, and we're asked how much leucine would we have in that. Couple of different ways to do this. Now, one thing you might be able to spot is that that is dividing by 20, so 1/20th of it. And then when we take our 15 grams of leucine, divide it by 20, that will give 0.75 grams. We have our calculator that we're allowed to use to find that out. Another way of doing it is if we try and work out what 1 gram of hemoglobin would have. First of all, to go from 100 down to 1, we would divide by 100. And then to go from 1 to 5, we would times by 5. And then you would do the same to the 15. And then that would give you, again, this 0.75. Now, that one takes a wee bit longer in this particular situation. But if you had numbers that didn't sit as nicely together, so if we were going from 10.7 to 3.3, there may well be a number that you can divide 10.7 by to end up at 3.3, but it's not obvious, certainly to me. In that situation, dividing by the first one to work out what 1 was, and then timesing by the second to scale it up the way would work regardless of how nice the numbers are. Next one, we're asked about a percentage success. This is when we have to calculate a percentage. To do that, we need the fraction, and in this case it's going to be the ones that were successful over the total number of cells, and then times that by 100. This would be how you calculate your score you got in a test as a percentage. So if you got 15 out of 20, you do 15 over 20 times 100. In this one, there was 210 successful and 350 in total. Times that by 100, and that gives you 60%. If they wanted to be a bit sneaky, they might have asked us for the unsuccessful percentage, in which case we could either go, well, if there was 60% successful, then 40% would be unsuccessful to take you up to 100. Another option you could do is, well, if there was 210 successful, that's going to be 140 unsuccessful. The difference between them, back over the 350 times 100 would have given us 40%, just in case they tried to be sneaky. Next one, we're asked about the build-up of a chemical in animal cells during fermentation. Fermentation happens when there's a lack of oxygen. In aerobic respiration, where there is oxygen, you would make carbon dioxide and water. In animal cells with a lack of oxygen, we make lactate. Sometimes we'll refer to it as lactic acid. Answer B is there because that's what plants and yeast make when they ferment. And then glucose is there because it's used during respiration. And then plants during fermentation would make ethanol and carbon dioxide. Or if it was aerobic respiration, cells would make carbon dioxide and water. So it's just trying to jumble up those kind of things for us. In question 8, they're talking about temperature affecting fermentation. We've got 3 different temperatures we're looking at, and we're counting the bubbles produced and counted. Which one should be kept constant during the investigation? This is trying to make a valid comparison, but the only thing we want to change is the temperature. Well, they are changing the temperature and using 3 different temperatures. So that means then that A and C will be incorrect because that's what they're investigating. What would have an impact on these? Well, yeast will make ethanol and carbon dioxide. That's that bubbles that are being produced. You can't keep that the same. We can only measure it and count it as it's coming out. But if you leave them for the same length of time, you'll be able to compare the results better. If you had one that was left for an hour and one that was left for a day, you'd expect the one that was left for a longer period of time to have just made more bubbles. Anyway, if we keep the time the same, you can compare them better. I think I said that already. During mitosis, what happens immediately before the cytoplasm divides? Well, going through the list, the very first thing that needs to happen is the chromosomes thicken and shorten. They then line up at the equator. The pairs of chromatids are pulled apart by the spindle fibres. Then the nuclear membrane forms just before it breaks apart. So the cytoplasm dividing is the end stage, which gives you the 2 daughter cells. In this one, we're talking about interneurons. In the pathway, we go the receptors of the sense. We go sensory, inter, motor at the end. Well, motor to sensory is the wrong way around. Effector is the part that's controlled by the motor neuron, so that might look like a muscle or a gland releasing a hormone. It's not going to be that way around. Sensory first, then motor sounds nice, whereas the other one is something that happens, but that's not the purpose of the interneuron. Talking about glucagon. Glucagon is required when there is a decrease in glucose, when the glucose is gone. It's produced by the pancreas along with insulin. Both A and C, they're both targeting the liver. So that's the same. When the glucose is gone, we want to make more glucose. That is the conversion that glucagon stimulates. If we're talking about insulin, insulin would be made by the pancreas. Insulin would target the liver and causes glucose to be stored as glycogen. So insulin happens when there's an increase in glucose and it reverses that. Here, less glucose, so this makes more to replace that. Here we're predicting how many people will have diabetes in year 10 if the average yearly increase continues. So we'll go through the numbers. We've got 6,000 more. We've got 7,000 more. We've got 7,000 more. We've got 6,000 more. Okay, I'm going to try 2 things here. So again, we've got the years. Now, if I just add on, let's say 6,000 each time, so that would be 310,000, 316, 322, 328, 334. This time, let's look at what happens if you add 7,000 on every time. So we would have 311,000, 318, 325, 332, and 339. As they're talking about, it's an average yearly increase. That means that the value added on would be somewhere between 6,000 and 7,000. And if we look at our two values, the only answer that fits within the range of my options here would be B for 336,500. I'm now spotting here what that's probably what to do is to find the average of the four increases, add them up, divide them by the number of numbers. And if we do that, we would get 310,500, 317,000, 323,500, 330,000, and then 336,500, which is exactly what they have as their answer there. So my first approach lucked out, I suppose, because I had options and there's only one option that fitted within my two values. But if we're asked to get the exact value, we would want to be using the average of the increases. Moving on to talking about the number of eggs produced by the common frog. What are they saying here then? Number of eggs produced by them. Okay, so here we're doing a reverse percentage where we are told that 5% survived, and we are told that that equals 23 eggs. And what we want to do now is we want to work out all of the eggs that were produced by this frog. So whether the last one. If we want to go from 5 down to 1, we'd divide by 5. Then from 1 to 100, we would times by 100. And then if we do the same to 23, that gives you 460, making our answer D for that one. In this one, we could have just times by 20 because we've used the same scaling that we had in the previous one. If you're ever unsure about what you have to do with the numbers, well, we can try it out with the example ones that they've given us. So if I follow the same process for the trout, well, 9% is 270, and I want 100%. Divide by 9 first, times by 100 second, and now do that to 270. And what I'm hoping to see is 3,000, and that's exactly what I get. So that's making me confident in what I've done. In this one, we're asked for the average monthly increase in the length of the fetus from month 5 to month 9. Now run it up and get our two values. Okay, we're going from, it's halfway between 200 and 300, so that's 250. And we're going up to, well, every box is going to be 10, because then there's 10 boxes to give you 100, so that makes that 410. To find the increase, we take them away from each other. That gives you 160 millimeters in, well, that's 1 month, 2 months, 3 months, and 4 months. And then to figure out what 1 month is, well, to go from 4 to 1, divide by 4. Same thing you do on the other side, which is going to give you an answer of 40 millimeters, which gives us B as the correct answer. Okay, moving on, we're now dealing with the different alleles and the inheritance that we'd see. Hetero means different, so that means that we're going to have one of each allele, and I'm going to use C. I just need to be careful that you can see it's big C, small c. So we have big C, small c, big C, small c. We can have 2 big C's, one of each, or 2 small c's. What I would then do is let's go back and look at, well, what do all those things mean? Well, they say that short coat is dominant to the long coat. So the big letter is going to be short, and then the one with 2 little letters is going to be long. And then in the middle, and you've got one of each, the dominant one would be expressed. That means they would also be short. So if we are looking at short versus long, there's going to be 3 short and only 1 would be long. And remember, this is the ratio of it. So we have got a total of 72 offspring. There are 4 parts in total. So to find out what one part would be, we want to do the 72 divided by 4. It should hopefully be 18. So if there's 18 long, there's going to be 3 times as many. And 3 times as many would give you 54. That gives us an answer of C. Another option is you can start putting the numbers in. You need to make sure that whatever number you have for short is 3 times as big as the number for long, and they add up to 72. That's the 2 things that are important. In this one, we're dealing with the heart. Yeah, when dealing with the heart, we have to remember that the sides are the opposite way around, because it's as the doctor or medical professional is looking at the person. So the one that we would say would be on the left, this one here, is the right. Atriums are above the ventricles, like an attic is at the top of a house, and then this will be your right ventricle, and then you have your opposite one, so left atrium and left ventricle. So they are being told we are starting off in the right atrium. We're going to go down to the right ventricle. We're going to go out to the lungs, as we can see from this diagram, and the vessels to do with the lungs are called pulmonary. The vessels going away from the heart, so it's the pulmonary artery, comes back through the pulmonary vein into the left atrium, down to the left ventricle, and then it would go out to the body. From the ones we want, A is correct for number 16. Next one, we're dealing with reliability. In this one, we are looking to do repeats of the experiment. Well, increasing the rest period is not going to increase the reliability. If you do it in a different location, that's you making the experiment less valid. You're changing some more things. Again, changing them, it's not going to be helping with reliability. Whereas D, you're repeating. So that could be, in this case, repeating with lots of different people doing the same exercises, or you could make each student do it multiple times. This might be something you came across when doing your assignment prep. Repeating for reliability. It's not accuracy, it's reliability here. In this one, we're asked about sewage works and samples of water and the oxygen content. What is the percentage increase in oxygen content from 250? Okay, what must we be going up in? Now, I'm going to hazard a guess at 0.02, because that would be 0.04, 0.06, 0.08, and then 0.1. So 0.04, let me ping that in first. If you're ever unsure, one thing you can do is if you take the gap, so in this case I'm just going to go for the first section, and then that is split up into 5 little boxes, and when you do that you also get 0.02. Sometimes the numbers are quite nice, sometimes the numbers are a wee bit more obscure, sometimes the numbers are a wee bit smaller, makes it a wee bit more difficult. Let's go to 1,000 now though. And then here we're going up to 1.0. Now it's going to be a big percentage increase because it's a big jump. How do we do it? We want to have the difference over original times 100 or doof. Take them away from each other, you get 0.96 divided by. When we're talking about an increase, we're starting at the smaller one and going up to the larger one, going from 250 to 1000. So we're using that first value of 0.04, times that by 100 to turn it into a percentage. You end up with 2,400%, and that makes C the correct answer for that one. When it comes to just dealing with percentages of a whole, 100% is the maximum, but percentage increases can be over 100%, as we have got here. In this one, we're talking about types of competition. Well, like an international flight is between countries. Intra is inside. Seals and dolphins are different species, so that means it's going to be inter specific, and they are competing for food. The food is something that's living, that makes it biotic. That's going to put our answer as C for number 19, just like that. In this one, we're talking about photosynthesis. We're asked to compare 2 tubes to show the effect of light on photosynthesis. So the only difference should be how much light there is. Let's go through our options. So 1 and 2 is the first option. The only thing that's different between 1 and 2 is the carbon dioxide concentration. Next up, between 1 and 4, what difference have we got? Well, there's a difference in carbon dioxide concentration again, but there's then also the difference in light. That's not the one we're going to pick, because how do we know what is causing any changes? You can't. 2 and 3, it's again both. So CO2 concentration and light. So that must make it 2 and 4. Just double check. Same concentration of carbon dioxide, only light is different. That'll make D the correct answer for that one. In this one, we're looking at limiting factors at 2 different points in this graph. Well, at point X, as we go past point X and increase the light intensity, that is having an effect. That means that light intensity was preventing the reaction from running as quickly as it could. So X is light intensity. That leaves us with temperature or concentration. Now, just using logic, we can rule out the temperature because it doesn't actually tell you anything about temperature there, but it does tell us about carbon dioxide. The other thing we can do is, if we are looking at this point here, if you compare it to the point directly above it, the change that we've made is... Oh, did I mention temperature? Okay, the temperature's the same in both of them, so changing the temperature has made no difference to it, but adding more carbon dioxide has made the rate of photosynthesis higher. That means carbon dioxide was having an effect, so B would be the correct answer there. In this one, we're talking about the pyramid of numbers, so how many of each thing we've got. You have lots and lots of grass, which makes the bottom bar have to be wide. You'll then have less rabbits, less foxes. So the ones that are looking good for me just now would be those two. This is more going to be if there's like one big tree supporting all the animals. It's not like that. And one fox could have lots of fleas on it if it was flea ridden. Is going to be the correct answer for this one. Shuffling on, we are asked about what happens to the percentage of owls with brown feathers, which is just what the graph is showing. Find our two values for point 1, 1993 and 2000. So 1993, that's at 21. And then at 2003, we're at 42. I mean, that does look like it doubles, which would suggest that's going to be correct. We'll have a look at the other ones. So B is saying it's continuously increasing throughout the whole period of the graph. Well, that's not right, because it's going down, decreasing there. Increases more at a certain point. So we want to compare 1888 to 1993. Oh, so they're saying that's the biggest increase. Now, to be the biggest increase, it would need to be the steepest section of the graph going up the way. It is steeper than the 1993 to 1998. It's less steep than the 1998 to 2003, meaning that that statement is incorrect. And then they're saying it increases less. So they're trying to say that this one here is the least steep. But if we go to that last block, we can see that the last section is less steep. Therefore, that would be the slowest increase. Goes to that. Right, this one, we are using animals or insects, organisms to eat pests. That is described as biological control. Genetic modification, you would change the DNA, the genetic information, which is not happening. And C, you'd use chemicals to kill off the insects, which is not happening. And fertilizers, they improve the nutrient level, which is not what we're doing in this one. In this next one, we are asked about the percentage of moths counted in year 4 that were dark. Okay, so percentage, like the test score, we want to see how many were dark. Year 4 was 180. And then we want the total number of moths. So in this one, what we have to do is add both the values together to get the total moth population. And again, we want to times that by 100, gives you 60%. That's going to make our answer D. Moving on to section 2. Here we have to look at the different structures that would be found in a bacterial cell. Right, bacteria will have a cell wall. It's animal cells that don't. Nucleus is found in everything except a bacterial cell. Bacterial cell have the loop of DNA and plasmids, so it's not that. They do not have mitochondria. They don't carry out aerobic respiration. They can make their own proteins in their Ribosomes, they do have plasmids, they need a cell membrane, and they will not have a vacuole. Here we're asked about plant cell walls. They are made up of cellulose. One of the things that can happen when plants make glucose in photosynthesis, they can store it as starch, or they can use it to make cellulose for the cell walls. Here we need a whole number ratio. So let's put the numbers above. So that, and that. What can they all get divided by? Divide by 100. And because we need to keep it as a whole number, any other number would make some of them be point something. That's just going to be the answer there. Muscle cells require lots of mitochondria. The mitochondria carries out aerobic respiration, which releases energy from food, produces ATP. And then muscle cells require lots of energy, ATP for moving, so for contracting. And then previously they were asking about comparing muscle to skin. Same idea that muscles need to move, contract, so they need more energy. Next one, we have a bar chart. Name the ion that moves from seaweed into seawater by diffusion. Diffusion is from high to low. So we are looking for something where it's higher in seaweed and lower in seawater. Oh, so comparing first two bars in each one. Sodium is lower in seaweed than seawater, so it's not going to be that. Potassium is higher. So that's going to be the one there. And if you look at chloride, you see chloride is also lower in the seaweed than the seawater. So chloride and sodium would do the same thing. Next one, they're looking for a sodium ion concentration below 0.25. And they're trying to move excess sodium out of the unicellular organism. Okay, sodium, unicellular. So here. And it's going to move it out into the seawater. And what can we say? We're moving from low to high. That's the opposite of diffusion. That is active transport. They're telling us it has to go out, low to high, active transport. And then cell sap is found in the vacuole of cells. Just like that. Next one, we're talking about enzyme activity. Breaking up sucrose into glucose and fructose. Identify the temperature at which invertase was most active. So the higher activity was 45. So just using the key. It's higher up, more activity units. Just 45. And then optimum pH is the pH where the activity is the highest. And that in both of the temperatures happens at pH 5. That'll be that. Okay, next on, what would happen if we did 75? Enzyme activity would decrease. Might even drop to 0 because the enzyme would be denatured and then the substrate would not longer fit in the enzyme's active site. Or something like that. How could this be done? Find the more exact one. So if we want to dial in the exact best temperature, we would want to repeat the experiment with more temperatures around that ballpark region. So we've got, as it sits, the 45 degrees has worked the best. When it gets to 55 degrees, the activity has dropped. So we want to test out values that are between those two numbers. And that should let you narrow it into that one. Why would invertase not work in this? So the active site of each enzyme is specific to one substrate, and this is not the enzyme for this particular reaction. Here we're talking about genetic modification. What is a gene? This is one of the definitions we need to know. Section of DNA that codes for a protein. So one use of enzymes, they are used to extract the desired gene. So that would be to get the human gene out of the DNA. Another thing it does as well, it would cut open the plasmid of the bacteria. We only need one of these, but I'm just going to write them all. They're also involved in sealing the human gene into the plasmid. Right, another proportionate one. We have got 45,000 litres of blood produces 400 grams of interferon. And we're wanting 1 gram of interferon. To go from 400 to 1, you can divide by 400. So do the same to 45,000. And that will give 112.5. All right, so there's 85 milligrams is produced by 1 litre. And we're asked about 45,000 litres. How do you go between them? Times by 45,000. So 85 times 45,000. And that gives 3,825,000 1000 milligrams. And then to get our actual answer, we're asked about grams. Well, 1 gram is 1000 milligrams. So if we've got this many milligrams, we need to divide it by 1000. 3825 grams. We could have divided the 85 milligrams by 1000 to run into that many grams. We then times that by 45,000 and we still get the same answer. Another type of protein produced by the immune system, that would be antibodies. Okay, this one we're talking about breaking down glucose in animal cells. This is aerobic respiration. Okay, what happens? Well, first we start with glucose. Glucose is broken down into pyruvate, and then pyruvate is broken down into carbon dioxide and water. This would be the same as if it was in plant cells or yeast cells with oxygen present. We're also making ATP. Other things you can talk about is where they happen. So stage 1 happens in the cytoplasm, and stage 2 happens in the mitochondria. That'd hopefully be enough for all 4 points. And this one we're on to the nervous system. We're asked about the central nervous system, that is the brain and the spinal cord. And then we're asked about the large part at the top of the brain, that is the cerebrum. We also have the cerebellum. And then the one they're asking us about, which is the medulla. Cerebellum is for balance and coordination. Medulla is for regulating heart rate and breathing rate. Receptors pick up the information that's coming in to the body, detect stimuli. So that might be like touch, smell, taste, all those kind of sensory things. Next one, asking about reproduction in humans. Sperm is produced in the testes. Cell A, which is the zygote, the first cell that's produced when the sperm and the egg join together during fertilization. Sperm has half the information, it's haploid. So it's one set of chromosomes, whereas cell A would be diploid, because it's got 2 of them. Cell A would be diploid. Here we've got cells dividing, cells growing into more cells. So mitosis or cell division. Why are stem cells important? Stem cells are undifferentiated, unspecialized cells that can grow into lots of different cells. So I'll probably go for unspecialized. Or question 8, we're dealing with dominant and recessive characteristics. They're saying that the ability to roll their tongue is dominant. They can have 2 dominant alleles, they can have 1 of each, or they can have 2 little ones. Because the bigger is dominant, that means that anyone that's got 2 of them will be able to roll their tongue. If you've got 1 of each, again, you still be able to roll your tongue. And then the last one will be a non-roller. So they are saying that we've got a male with that. Oh, okay, so child is unable. So the only way to get that, because the non-rolling is a recessive characteristic, they'll need to have 2 little letters as their genotype. Next off, we're looking at hetero or homo. They have 2 of the same. We use homo in science to describe that. They have 2 copies of the same allele. Alleles being the different versions of the gene, big R and small r. In this case, the male and the female are having another child, and we have to do the percentage chance that this child will be able to roll their tongue. We have got capital R, baby R, baby R, baby R. Going through the combinations. 1 big, 1 small, 2 small. 1 big, 1 small, and 2 small. Will be able to roll and use our wee reference at the top. That means they have at least 1 R, which in this case would be. So in this question, we've got 2 of them can roll their tongues, 2 of them wouldn't. That is a 2 and 4, 1 and 2, a 50% chance of doing that. In types of variation, we have discrete and we have continuous. Discrete, we fit into neat little boxes. They can roll their tongue or they cannot. Continuous with more things like height, where there's a range of values that it could be. This one's discrete. Moving on to talk about transpiration, which is water lost by the plants. The openings found in the leaves, that is the stomata. Which plant was used as a control? So the control would be the one that's left without doing anything to it. And that looks like plant 1 because they're checking the effect of the petroleum jelly. So that's looking like plant 1 is just left to see what would happen if nothing changes, and then we can compare plants 2 and 3 to them. Why is it necessary to cover the plant pots? Well, that prevents evaporation of water from the soil because transpiration's through the leaves, so that's what they're looking for. That if you hadn't done that, then the water loss would be twofold from the leaves and the soil, and you couldn't tell exactly how much was from each of them. Shuffling on. Why are we doing it as a percentage change? If you look at the starting masses are all different, so you would expect, for example, the bigger plant to have lost more just in general because it's got more to lose. But if you do it as a percentage, it allows a comparison between them. Okay, now we have to answer our aim. The aim is to find out which leaf surface loses most water through transpiration. Okay, so we're asked which leaf surface loses the most water. So we're looking, is it the upper surface or the lower surface? Well, if we do the comparison between the non-covered and then plant 2, there's only a 4% saving of water, which means that there was only around about 4% loss in the upper leaves, whereas on the lower leaves, we have now saved ourselves 36% water being lost. So that means that was the lower surface. Yeah, that has more of the stomata. That makes sense. So lower surface loses most water by transpiration. On the next one, we're on to the blood. We are looking to check if the patients have the components within normal range. So let's do our comparison. I'll highlight green if they're normal, and then we'll do red if they're abnormal. So 6.5 does fit in that category. So does patient S. Next one, that fits nicely, but patient S does not. Now at this point here, the answer has to be R, because as soon as they have one that falls out with it, they're ruled out. But let's just double check. That is banging between. There's a second one that's out of the range. Fine and fine. So it's only patient R who has everything in range. Next one. Both patients had their blood retested. Patient S had increased by 20% hemoglobin. So patient S was 115, and then that's going up by 20%. What could we do? So one option would be to say, right, well, that was 100%, and we're now trying to get to 120% because we're adding on another 20%. I'm going to stick in 1% here. If I divide by 100, that tells me what 1% would be. And then I want to times by 120. And because we have a calculator, we can just ping that straight in. And that'll give us an answer of 138 units. Another option is we could work out what 20% of the 115 is. Now your calculator might have the percentage button that you can use. Here I'm going to do it as 20 over 100 for 20%. Times 115, that gives you 23 extra. So you add that on to your 115 starting, and that again gets you 138, just about which way makes more sense to you. Next one, we're talking about one way in which a red blood cell is specialised, apart from hemoglobin. So I've dealt with that already. So some of the things about this would be there is no nucleus, which allows more oxygen to be carried. There's going to be more space for oxygen, allows it to carry more oxygen. Another thing is the biconcave shape, that kind of caved in at the top and the bottom, and that allows a higher surface area for more efficient transfer of materials, and that again allows more oxygen can be carried, and both of those things allow it to carry more oxygen. Another thing about the shape is it means it can be quite flexible, so that when it's passing through vessels it can squeeze its way through. Next up, we have to compare arteries and veins. There are 3 marks, which I'm going to say there's going to be 3 points. We'll compare the walls first of all. Arteries have thicker walls than veins. Arteries carry blood at higher pressure, that's why they need thicker walls. You're not asked about the blood that's passing through them, they're talking about structures, so physical things about them. Second point I'll make is that veins have valves and arteries don't. Last one I'll do is about the openings. So arteries have a narrower opening, a narrower channel, lumen, than the veins. And that also contributes to the fact that the blood goes through it more quickly. Like if you squeeze the ends of a hose or cover over with part of a tap, the water comes out faster. Has to be structural things. Make sure you do that for that one. Next one, we have a bunch of information about a study, and we have to answer some questions on them. Elite athletes have more tooth decay than general public. Why is that unexpected? Well, from reading the information, it says that they brush their teeth. More athletes brush their teeth twice a day compared to the general public. Reading through it as well, you could also talk about the flossing as well. Yeah, a higher percentage of athletes floss regularly. And another thing you can say as well, it talks about them putting in more effort. Is what the study shows for that one. They smoke less as well. But yeah, I'll go for that one there. How many of the athletes flossed regularly? It says that there were 44%. Does it tell us? Yes, it does. 350. So you want to work out 44% of 350. We can do 44 over 100 times the 350. And that'll give you 154 of the athletes regularly flossing. Why might it not be valid? Now, in an experiment like this, there's so many things that can affect the healthiness of the teeth that they're not able to control everything else. So the only difference won't be athlete versus general public. They might have pre-existing problems with their teeth. They might have not gone to a dentist to get the help because of something in their past. They might have fake veneers put in, which then would mean that they're not going to corrode. There's so many different things that can have an effect that it would be impossible to control all the variables except from, are you an athlete or are you not? Also doesn't say anything about age as well. So it could be the age, the genders of them. They're just grouping them together as athlete or not an athlete. So it's multifactorial. It's too complicated. There's lots of different variables, things that can have an effect of them. Too many factors to control. Moving on, we have got a sports energy bar, and we have to do a pie chart. It's out of 100. Now, what that will mean is if we go into the halfway line, that's got to be 50. So that's looking like each of these little sections has to be 5%, or 5 grams, sorry, has to be 5 grams. So if we're going through the fat is needing 4 boxes. So taking it from the middle, well, that would be 5, 10, 15, and then 20. And that is for fat. Next up, we'll do sugar. And then for this one, we want 5, 10, 15, 20, 25, 30, 35. Next up, we've then got protein. And if I'm doing this right, that will leave us with 2 boxes left for the other. Other will squeeze in there. 5, 10, 15, 20, 25, 30, 35. And then 5 and 10 for other. Just like that. Shuffling on, we're now into food web, which is showing the different interactions between different animals. We are looking for a producer. The producer is what's going to produce their own food. They are going to be a plant. They're going to be at the start of the food chain / food web. That will be pine tree or moss. And you just need one of them to pick up the mark. Next up, we're asked about a predator. Squirrel is eating a tree. That's not chasing after and hunting down and killing another animal. Whereas all the ones like the hedgehog, wood mouse are eating the earthworms, so they're the earthworms predators, and then foxes and owls eat the mouse or the hedgehog. So it's really anything except from squirrel, pine tree, or moss. Again, just one of them. Your owl and your fox are your typical predators that they kind of ask about in that one. Now we're saying what would happen to the size of the squirrels if all the foxes were removed. Look at the relationship between the foxes and the squirrels. Well, the fox eats the squirrels, so if the foxes were to be wiped out, then the squirrel population would likely increase because all the foxes are removed. So I can say no squirrels would be eaten by the fox. If it was just reducing some of the population of the foxes, then less squirrels would be eaten. But in this one, if there's no foxes, there'd be no squirrels eaten by the fox. Are there any arguments for another way? What else is in that? Well, if the foxes aren't there to eat some of the squirrels, the owls might have more squirrels to eat because there's less competition, so they might eat more of them. Or it might stay the same if the number of squirrels eaten by foxes is balanced out by the extra squirrels eaten by the owls, potentially. But I think the increased one sounds most likely. The role of an organism is its niche. That's all the interactions it has with its natural environment. So, for example, for a squirrel eats the pine tree and then is the food for the owl and the fox. We have to use our paired statement key to work out which fungus goes in which part of the key. So we'll go through the ones that we already know just so I can show you how it works. So sickener. So if you follow through that, question 1 is about the stem colour. So for the sickener, the stem colour is white. So if it's not dark brown, we go to question 2. And in question 2, it asks you about the cap colour. It's bright scarlet. That makes it the sickener. Done and dusted. Always start from the start. So they've already done the velvet and the pelargonium bonnet. Pelargonium bonnet. Let's now then look at the ear pick fungus. Lovely name. Let's go through and say, so stem colour, ear pick is dark brown. Go to question 3. Cap colour is dark brown. That puts the ear pick fungus in here. Okay, next we have the pinewood ginger tail. Its stem is brown, which is not dark brown, so we go to question 2. It has a bright brown cap. That means that has to get pinged in here. So pinewood ginger tail. Now something's gonna miss here because right, I've got myself muddled because the ear pick fungus has to go down here, because what I didn't catch was with the cap colour being dark brown, 2 of them have dark brown stem and dark brown cap colour. So to tell them apart, we need to go to question 4. So if there'd only been one of them, what I did at first would have been fine, but made a bit of a mistake there. So now if I do that, double checking going through, so just pick the pelargonium bonnet. You've got a dark brown stem, which means go to question 3. It's dark brown cap, so go to question 4, and it has a 1 centimetre maximum cap, and that does work out for that. So I just missed out that question bit in there. Moving on to photosynthesis, we have to do the average time, and we're going to just take all the numbers. So this is your mean. You're going to add up all 5. That gives a total of 78.3. Divide that by 5, and then we could round that to 15.7, the same way as they have 1 decimal place. That would get us our answer there. A gas produced during the first stage of photosynthesis. The first stage are the light reactions where water splits and oxygen is released. The hydrogen moves on to the later stage. So oxygen would be the correct answer there. Moving on, we're asked about the molecule generated by chemical energy used in the second stage. That is your adenosine triphosphate, or ATP. The second stage is carbon fixation. So the carbon dioxide is going to get joined together. Why is it affected by temperature? Well, photosynthesis, respiration, they're all enzyme-controlled reactions. As we see in the previous question, enzymes activity can be affected by temperature. Next, we're on to sampling in a woodland. This is a pitfall trap where the animals or where the insects will walk along and then fall down it and be trapped. If we're comparing different pitfall traps, what needs to be the same? So I suppose one of them would be the size of the trap, the depth of trap as well, to make a fair comparison. The camouflage, I suppose, if you're putting a couple of leaves on top, you want to do that every time. You can't have some open and some covered over, because that would be an unfair test. How to make it more reliable? Repeat for reliability, set several traps. So multiple traps means you can then get an average of your results, making it more reliable. In this one, we have a pyramid of energy, because all the values are energy. 5% of the light reaching the plankton is converted into new plant material. Calculate how much energy it received. Right. So they're not asking us for what 5% is. Kind of sounds a wee bit like that, but it's not quite. They are saying that the light that comes in, they're saying that this, I think they're saying this, is 5%. So if I'm right, that would mean that 5% is 42000. And we want to know all the light totaled. So again, let's stick with what works. We could times it by 20. Seems to be quite a common scaling thing for this paper. But I'm going to divide by 5 times by 100. 840,000 units. There. Ways in which energy can be lost. That could be through movement. That could be through heat. Or that could be undigested material. Next on, we've got different wrens developing off the west coast of Scotland. They are thought to be evolving into new species, but they're trying to prove that they're not yet a different species. You would check if they can interbreed to produce fertile offspring. If their babies can have babies, then they will be the same species. If not, then they'll be different species. We have to put this in the correct order. They've already told us A is the last thing to do. And the first thing they have to do is there has to be some sort of separation or isolation on an island. There's then going to be changes in their DNA. That's going to be a mutation that's going to happen. You're then going to have natural selection, where because of the conditions in that area, one of the mutations is favourable, so they will survive and be able to pass on their genes, and then over time they will become more and more different. So lots of rounds of mutations, selection will get to two different species. How does natural selection result in the evolution of species? So it's survival of the fittest. So the best suited individuals survive and reproduce. And then your favourable or advantageous alleles are passed on. That brings us to the end of the question and the end of the paper. Thanks for watching. Goodbye.